Good morning, everybody. I'll just repeat what I said just a minute ago because there were a few other people on Zoom that just joined. So the way today is going to go, today is an introduction to single-cell RNA-seq and some steps for data pre-processing. We have 2 hours where we're going to talk about some of the biological rationale for why we even do this and how we go about doing the experiment. And then there will be a live workshop for everybody, both in person and those on Zoom, to go through a sort of basic pre-processing workflow using a tool called AlvinFry. There's going to be a few periods of time where we're going to be like downloading something or waiting for a tool to run. So those would be good times to kind of stack some questions, like if you're having some trouble with an install or something like that. But during this first section where we're talking about biology, like feel free to interrupt at any point if you're in person. We have some people fielding questions over Zoom in the chat as well, so you can put your questions there. As a very first step, we're going to actually get a jump on one of these things. So because we're going to use reference genome and reference gene annotation, the very, very first step that we're going to do, because this download takes a good 20 minutes, is download the HG38 or GRC H38 primary assembly reference genome, as well as the GenCode GTF. I'll explain what these things are if these terms are unfamiliar to you. If you navigate to gencodegenes.org/human, you'll be able to copy and paste these very, very long URLs. A couple other notes. One is there will be code on my GitHub. The slides will be there. Everything is being recorded, so all of this will appear on YouTube later. So if you miss something as we're going through it, don't panic. We will get you up to speed either during the session, or you'll have plenty of time to kind of go through all of these details after the session wraps. Let me just quickly show you where things are on GitHub at the moment. So if you go to jeremy-sim.github.io/ds_scRNA_2024-0228, and then the same thing .html. There's a nice ready to go HTML markdown document with a lot of links, all of the code, all kinds of stuff. And somewhere in here, yeah, maybe I didn't include the reference genome annotation. Oh no, it is here, thank you. Yeah, retrieve reference genome and gene annotations. The links are all here, you can copy and paste.
Both of them?
Both of them, yeah.
Okay.
Sometimes if you're using a Mac especially, this tool called wget is not something that is pre-installed. So you may get an error straight away that says, I don't know what wget is. You can swap that out for Perl-O instead of wget, and then the rest should work. So we want to get these downloads going, especially the reference genome takes a good 20 minutes. So we can talk about some of the background while this is going for us. But you'll need both of these, the genome FASTA, which is really just every single chromosome in the human genome, every single nucleotide, as well as the GTF file, which is an annotation format that contains all of the genes, exon coordinates, every last detail about gene annotation built on the HG38 reference. On the slide, I have some notes here too. The data set that we're going to be working with is human derived. If you're here because you're interested in doing something for another species, mouse or otherwise, obviously you're going to need a different reference genome and annotation for those projects, but today let's just focus on human HG38. Anyone panicking on Zoom or in person with the download before we get rolling? Now would be a good time to raise your hand or ask a question. Okay, let's hope that everyone is underway and we're going to get rolling. So as I said, today is an introduction on two fronts. One is the biological rationale for doing these types of experiments, and the other is a live demo on running some data pre-processing, starting with fast tissues that are publicly available from 10x Genomics, and we will process those into a cells by genes data matrix using a tool called AlvinFry. So we'll start off with some rationale why we even do this in the first place. And then we'll go through an overview of a variety of experimental methodologies and platforms. 10x Genomics is one of them and arguably the most common or popular, but there are some others that, that do have some very good use cases. So I'll talk about some of those as well. I'll go through some best practices for experimental design. Many of the people here are in the data science department, whether they're computational biologists or biostatisticians. And I think it's really important to get people like us involved at a very early stage so that we can help design proper experiment so that we get the best possible data after the experiment is run. I'll also give an overview of some of the data processing tools. Today, we're going to use AlvinFry, but many people use 10x Genomics' software called Cell Ranger. And I'll just briefly talk about some of the differences there. And then the bigger chunk of today is going to be a live demo pre-processing using AlvinFry. So as I mentioned, all of the slides, all of the code, the recordings, everything is going to be available afterwards. So if you're totally lost and just need to, digest for a little bit longer, you can re-watch this at your own pace afterwards. And of course, feel free to reach out to me at any time after this by email or over Slack or. Okay, so why do we do single-cell RNA-seq in the first place? So as hopefully everyone knows, complex tissues contain a lot of diverse cell types at different proportions. So on the right, I just have a cartoon representation of a chunk of tissue. There's a bunch of different cell types. There's the green cell type, orange, blue, and black. And if we were to profile this tissue in bulk, that would mean just taking this entire chunk all at once. And if we did bulk RNA-seq, the result would effectively be a population-wide average of all of these different cell types. So this toy example here, I have 5 different genes and their counts for each of these different cell types. If we were to sort them magically separately, we would know that gene A is expressed at a level of 10 in the green cells, 5 in blue, 50 in black. But for all of these, if we just took this population-wide average by doing bulk RNA-seq, we would effectively take the average of these values. So our tissue expression would look like this on the right, but really it is a representation of all of the different cell types underneath that average. And if you take 2 different chunks, the composition could be different, and therefore your average can be different. So especially when we're comparing across conditions, I just labeled them as condition 1 and condition 2. They could be wild type mutant, could be tumor normal, it could be all kinds of things. But you can often, when we do comparative analyses, we're looking at things with a change in the tissue composition and the cellular proportions within that tissue sample. So here, this first scenario, these tissues are identical, except for the fact that cell type D in black is far more prevalent in condition 2 than condition 1. Another scenario is that the actual counts of those cells are the same, but somehow functionally they're a little bit different. So here I've labeled these black cells with a black outline indicating that cell type D prime has some kind of altered cell state, even though the physical number of cells is identical. And that could mean there's an altered gene expression in a subset of cells, or it could be any number of other things. So in either of these scenarios, if we were to do bulk RNA-seq, we likely would have insufficient power to detect either of these differences. Just because it's a population-wide average, it's going to be just a mess, basically, of everything that's underneath here. And the true effect is going to be diluted because it's a representation of many, many cells, millions of cells in most cases. You could also think of a scenario where an experimental lab might want to sort these cells based on their identity first, and then do bulk RNA-seq on these sorted populations. So here are these three conditions again. Condition 1 is the kind of normal. Condition 2, scenario 1 is we have more black cells. Condition 2, scenario 2 is we have the same number of cells, but that altered kind of phenotype of them. So we could use a method like FACS and split out our populations. We can count them, and perhaps by FACS, we would see that in this middle scenario, we have more black cells here than we had over here. Again, with counting, we're not going to see any difference just by literal counting of cells in this scenario, but maybe we could do RNA-seq on the sorted black cell population, and maybe there would be something different there. But you can see that this would get completely super expensive, very time consuming. It also basically requires you to have selectable markers, and you have to know what the cellular composition is to really divide out this population into all of the different cell types that comprise it. So for really complex tissues, you have 20, 50, 100 different cell types. It's completely cost prohibitive to do something sorting based as an approach. So here came single cell RNA-seq using droplet microfluidics, was one of the first and then became one of the most popular approaches to do this. So this initially came out as a method that was called DropSeq from the McCarroll lab. And this little animation is from their lab website on this. What is inside the 10x Genomics box is more or less this exact technology. So in effect, we co-capture cells with barcoded beads. The beads are typically, they have an oligo DT so that there's going to be some kind of poly-A-based selection. After sequencing, we basically are labeling all of the transcripts, and we can tell which transcripts came from which cells. The 10x machine has a few inlets. These are kind of like lanes, and each one can process about 10,000 cells at a time. But by default, only one experimental condition can be processed at a time. So I'll talk about some implications of this a little bit later. So here's some more from the McCosco paper back in 2015 describing this initial technology. I'm not going to go into the super nitty-gritty details here, but you have these barcoded beads. There's a PCR handle, a cell barcode, a UMI, followed by an oligo DT. And commercially, they generate these beads using like a barcode synthesis, basically, in rounds. After 12 rounds, you get kind of a growing shell around these beads that have nucleotides that have been synthesized going outward. And then there's all kinds of different combinations of unique molecular indexes, UMIs. And then from the experimental side, you have to kind of blow apart a complex tissue into all of its individual cells. So there could be some digestion or sorting or Bicol gradient. There's all kinds of methods to do this, and it'll depend on whether your tissue is brain or liver or heart or kidney or any of those things are going to behave quite differently. So the exact protocol is going to vary based on your tissue of origin. The actual microfluidics part looks somewhat like this. So it's a multi-chamber channel, as you saw in that animation, where materials flowing from left to right. In one channel, there's all of the barcoded beads. In another, your single cells come in. And then in these oil droplets, at some frequency, they will get co-captured one and one. So the ideal case is you have this little oil bubble. Inside there is a cell and a bead. And hopefully no other cell and no other bead, although that does happen at some frequency. You're going to also have lots of empty droplets, and that's expected. There's also going to be some ambient RNA material that gets picked up in the process that we can talk about later too. So with all kinds of complicated molecular biology, you effectively generate a library where you have all of your transcripts, and ligated onto those transcripts are these molecular indexes, the UMIs, as well as the cell barcode, which tells us which cell those transcripts belonged to. Again, those barcodes are unique to the bead. And so when you co-capture one cell with one bead, everything in that cell gets labeled with the barcodes that were associated with that bead. After sequencing, you effectively get something that looks like this. So you have your cDNA. We sequence maybe 50 or so base pairs of that. But every read starts with a cell barcode and a UMI. And so computationally, we can split that apart into all of the UMIs that came from each individual cell barcode, and computationally generate a data matrix that looks like this. So you have all of your genes, 1 through M as rows, and all of your cells, 1 through N as columns. And the cells here are going to be each of these cell barcodes. Generally speaking, the data processing workflow looks something like this. Is we have to kind of deconvolute the cell and molecular barcodes. There's going to be some sequencing errors and some other weird synthesis things that we want to try to correct for. We want to allow a little bit of mismatch, like usually one base pair mismatch. Then there is some primer sequence on the 5' end, potentially poly A sequence on the 3' end that we want to trim off. We have to figure out, so now we have a cDNA sequence, we have to figure out what gene that sequence was derived from. So there's got to be some kind of alignment step either to the whole genome or to the transcriptome. Determination of which reads came from which transcripts, a selection step of what constitutes a real, quote unquote, cell versus things that are obviously debris or noise. And then the end result of most of these methods is some kind of cells by genes matrix. This is what we would use for any kind of downstream analysis. So let me talk about some of other possible platforms. So this, what I just described, is how 10x genomics works. There's another high throughput method, initially called split-seq, and then they formed a company, and that method, and the company is called PARSSPO. And this method is nice for a few reasons that I'll get into, but it all works in a plate-based setup. Instead of a machine, there's no microfluidics, there's nothing complicated like that. It's instead just crazy complicated at the molecular biology level. Through serial rounds of splitting and pooling and resplitting and repooling your libraries, you gradually add on one barcode, and then a second barcode, and then a third barcode in serial. And so instead of just a barcode belonging to a cell, you actually get a combination of barcode 1, barcode 2, and barcode 3. And that unique combination is unique to a particular cell in your experiment. There's a fun, very accessible movie that they released on YouTube, and you can pull this link sometime later if you want to watch it and understand this method a little bit better. The benefits here are that it can process hundreds of thousands or even millions of cells simultaneously from multiple conditions. So if you think about, you want to test wild type and mutant each with like a treated, untreated, or dose 1, dose 2, dose 3 kind of setup, that's a lot of conditions. That would be tens of thousands of dollars to do that by 10x sequencing, but much, much cheaper to do it with this type of approach. And because all of it is done in parallel, that means that you are minimizing the potential for batch effects across those samples. Another benefit here is that it's not only oligo DT primed, there's also random hexamers, which means your library is not only poly A selected, but there's also internal amplification, meaning we're getting reads from the middles of transcripts, not just the 3' end. I don't have the latest numbers, but the cost per cell is like on the order of a penny or two. So once you scale up, it is not prohibitively expensive to do a very, very large scale experiment. This method has also been shown to be compatible both with fresh cells as well as some kind of fixation, usually like methanol or something. You can also extract nuclei from your cells and process them that way. The other method that I have no personal experience in, but is worth mentioning, is SmartSeq. And I honestly don't really know a whole lot about this because I've never used it, but the main distinction is that you get truly full-length transcripts per cell. So if for some reason you're like really interested in, I don't know, alternative splicing or something along those lines, or can't get away with a 3' biased library, then SmartSeq may be more appropriate. These days, it's not only all about RNA gene expression, there's also multiple other single cell modalities, as well as what we call multiomics approaches, meaning you can get more than one modality from this exact same cell. One of the most common versions of that that's used these days is RNA plus chromatin accessibility using a TAC-seq. And so that effectively gives you a gene expression profile as well as a chromatin accessibility profile on the exact same cell. So you get two experiments, not quite for the price of one, but in the same prep. Another common one is called site-seq, and that is RNA plus protein expression looking at cell surface proteins. I don't know that this statement is true, but my feeling is that this method was invented for immunologists because often cell-specific markers like CD4, CD8 are not super reliably expressed at the RNA level or detected in single-cell RNA-seq. And so we can do something like site-seq instead and look at cell surface markers like those at the protein level and simultaneously profile the underlying gene expression signature of those cells. So this is a common one as well. We also can look at, for T cells, you can profile the T cell receptor repertoire using their TCR VDJ workflow. And that too is paired. So you get the gene expression profile as well as the TCR clonotype information on the same cells. There's also a whole other field of people looking at protein DNA interactions like transcription factor occupancy or histone modifications. That's a method called SC cut and tag. You could do that in conjunction with cell surface proteins. That's SE cut and tag pro. There's also all kinds of people doing spatial techniques, various combinations of all of these things. So you can do spatial RNA, spatial attack, and all sorts of things. So I just wanted to mention that there is a whole blossoming world of single cell methods out there. Okay, let's talk through some best practices for experimental design. The very, very first one that I'm going to tell everybody is that replicates are very, very important. Some people out there incorrectly argue that since you're profiling many cells from the same tissue, the argument is that those cells act as replicates for each other. That is just statistically not true. So the better approach, if you're looking at human tissue samples or doing experiments in mice or cell culture plates, whatever, you need multiples of those. Usually at least 3, ideally more than 5 or 6 per experimental group. The reason for that is that the variability in expression, the variability in our detectable cellular proportions per cell type, variability just because of the sparsity of the data, all of that is quite large. And so we need just more replicates to really profile that space and have sufficient statistical power to tell differences between our experimental groups. So as I said, I usually recommend labs do at least 3 replicates or samples per condition to capture and hopefully account for this variability. Honestly, the more the better. I have done some experiments in mice where an N of 10 per condition was really necessary. For each sample or replicate, you really want several thousand cells. We usually target at least 50,000 reads per cell. And really you need at least 10,000 cells total per experimental condition, but ideally even more than that, usually 20,000 per condition. There are some papers that look at the kind of saturation rate of all of this, and that's where these numbers come from. If you're interested in doing some kind of atlas scale study, like you want to document every last cell type, including really rare cell types in an entire tissue, then you're going to need way more than this. It's usually on the order of hundreds of thousands to potentially millions. And you can kind of do the math of figuring out how confident we're going to be in a population that's less than 0.1% of the entire tissue. You need a lot of cells to do that. The next is to do our best to control for batch effects. So as I mentioned, 10x by default can only isolate one biological sample at a time per inlet. And so what that means is if you're doing 10 samples, that's 10 completely independent experiments in a way. And so if you do sample 1 on Monday and sample 2 on Friday, then you kind of have a confounder there in the sense that those are not processed in parallel. If there were any differences in the environment or the experimenter or the reagent kit or anything along those lines, you can be introducing a batch effect that you cannot control for or correct.
There's a question in the chat.
Yeah.
Do you want me to read it out?
I can see it. Hey, Nabi, let me just read through. So when you refer to replicates, do you mean rerunning the experiment on the exact same sample multiple times or having multiple samples? For example, if you're using human tissue, do you mean running the experiment on the same slide multiple times or having multiple slides from the same patient and running each once? Right, so some of this comes down to the definition of what a biological versus a technical replicate is. There may be value in doing either or both of those. Human samples are a bit more of a challenge here. And so often you don't have the tissue available to do the kind of replicates that are statistically ideal. Let me give an example in mice, just because that's like a little bit easier to think about. So if we were just interested in comparing mutant versus wild type, that would mean 5 animals of wild type, 5 animals of mutant. That would be what I would consider a biological replicate. So those are completely independent tissue or cell isolations from individual animals. In humans, if you're looking at, I don't know, tumor versus normal, or tumors of one type versus tumors of another, that would ideally mean 5 samples from one group and 5 samples from the other. So 5 individual patients representing each group. As I said, it can get a lot more complicated than that when you have like, longitudinal biopsy samples or paired normal and tumor or things like that. But yeah, you really want to just capture all of the potential technical variability that there might be within an experimental group as best as you can.
Okay, so you're saying, do you think they should be doing technical replicates as well, or is the biological replicate more important than the technical replicate?
Usually, I would say the biological replicate is going to be more important and informative. There may be cases when a technical replicate is important, especially if for some reason you need to profile cells using different platforms. That would be a technical replicate of sorts, where like one piece of a sample is processed with 10x and the other is with PARSSPO or something like that. That would be important to kind of do those. Yeah, most of the time we're talking about biological replicates. But this again, like philosophically or theoretically speaking, if you just ran the same sample through 2 inlets, is that a technical replicate? And if you ask 10 people, you might get 10 different answers on that.
Okay.
So yeah, it's not super clear, but it can be valuable to increase your cell count per individual sample. And that would, in a way, be a technical replicate, but you kind of, in effect, would just combine them and not really treat them as replicates.
Okay, yeah.
Okay, yeah, so here's what I kind of summarized: separate isolations for different experimental conditions. It's worth mentioning that as of the last, 2 years or so, 10x and some others have come out with a method they call cell hashing. Another name for this is a hashtag oligo. It's abbreviated HTO. And essentially, this allows you to multiplex cells in each inlet, which statistically speaking is more ideal. So if you had 5 patients that were from, or let's say 6 patients, 3 from one group and 3 from another, you could hash those and run them all on the same inlet. And that will minimize some of the potential batch effects if compared to if you were to isolate them on separate days or something. This is these oligos are attached using an antibody or lipid-based method. And so it is worth mentioning that it may not work for every single cell type or tissue. Kind of requires an antibody that is ubiquitous and will recognize and bind to any cell in your sample, which isn't necessarily always true. There's a question on the chat that says, how much time does one run take? Good question. I think my understanding, which is vague, is on the order of a few hours. So often if you have a large experiment, you can't really possibly do everything all in the same day. So it isn't fast. Often what people do is process cells fresh, which means you have a fresh biopsy that you separate out into individual cells, and you run it over to the facility that's going to do the 10x prep all in the same day. And that's often how it's done. So yeah, it is pretty slow. So for those of us working with external collaborators, really try to push people to like, minimize these batch effects as much as we can. If you're doing a mouse experiment, like at least mix a wild type and a mutant hashed together in one inlet, or like if all else fails, like at least try to get some samples from both experimental conditions done on the same day. Simple things like humidity or ozone or the experimenter, all of those things can have an impact on the data that we get eventually. And so minimizing it in any way is going to be better than not doing anything at all. And as I said, like other solutions like split-seq or PARSSPO may be more statistically appropriate when you have really, really complex experimental setups. So if you're, as I said before, if you're doing like, mutant wild type mouse, treated, untreated conditions for each, or a concentration curve or multiple time points or something, do all of those in parallel and get everything done all on the same plate, all on the same day. You can do that with more than 100,000 cells and multiplex it in that way and minimize those batch effects. Okay, so some other considerations to think of. Because 10x is a microfluidics approach, there is some fear that a really, really large cell would just literally get stuck, physically stuck in the channels, because the channels are only 10 or 20 microns or something. This is a hard one to evaluate because you only get what you get on the other side. And so you often have no idea what you're missing. I think some of this has been explored, but it's worth mentioning, especially for people that work in the brain. Neurons can be quite large, astrocytes, et cetera, have a lot of projections and other kind of cellular structures that could be a physical barrier to properly isolating and sampling the cellular composition. Once you have all of the data, we're really only probing expression of the top 10 or 20% expressed genes per cell. And it's a somewhat random selection of those 10 or 20% top expressors. We have a ton of zeros. And so that sparsity of the data is just something that has to be properly dealt with downstream. If your collaborating lab is interested in genes known to be lowly expressed, and often many transcription factors are not very highly expressed, because of this, droplet-based single-cell RNA-seq like 10x may not be the most appropriate method. Some people will do all of this using whole cells. Others will isolate nuclei first. And it's just important to note that once you get into annotation of the cell types, cell types or nuclear types, I guess, those expression patterns can be different depending on which compartment the RNA molecules were isolated from. Not every RNA molecule is exported out of the nucleus. There is some compartmentalization there. So it's just important to keep in mind that your expectation of what a cellular gene expression profile might look like could be different if you're looking only at nuclei. And another note is that 10x offers two different chemistries. There's most commonly 3' chemistry, which is oligo-DT based, meaning it is 3' poly-A selected. But there is a 5' based method also. And this is the one that is compatible with their VDJ T-cell receptor clonotyping workflow. And so the data processing is different depending on whether you have 5' or 3' data. And it's just, you should know this upfront before you get started on any analysis, what we're talking about, whether it's cells or nuclei, whether it's 5' or 3' chemistry, what are the cells, what's the tissue, what are the conditions, all of this you need to kind of collect and understand upfront. Okay, when we get into the analysis, this is an abbreviation and a phrase that I use frequently. you're only as good as your annotations. So it's really important to use an appropriate, updated, and most importantly, constant reference genome and gene annotations for all samples for a given study. I think often labs will rely on a core to keep things constant for them, but also often, especially here, we have a lot of patient-derived samples. I myself am working on data derived from a clinical trial where those samples were collected over a period of 3 years. And as those cells get isolated, by default, the core just goes ahead and processes those using whatever tool and annotation they have on that given day, which means the sample processed at the very beginning may have very different data than the sample processed at the very end of the trial. So it's really important to just like take a step back and treat everything equally. And so what I do is just go straight to the FASTQs, the raw data that hasn't been processed, and create one static instance of my reference genome and my gene annotations using the most up-to-date at that particular time. You can use either UCSC RefSeq annotations or GenCode ensemble annotations. I tend to go towards GenCode. It's a little bit more comprehensive. It therefore gives the best possible chance for a read to align and be appropriately counted. GenCode annotations are updated every 3 months, about 2 or 3 times a year. There's also a ton of discussion these days about whether a read aligns to a splice junction or an intron or an exon, and how to appropriately count for each of those different cases. You'll sometimes see phrasing that's spliced, unspliced, and ambiguous. We'll use this phrasing later. It's called USA, but there's also some other terminology that other labs use. But it all kind of relates to the same problem, is if a read maps to a splice junction, then we have evidence that that splicing event happened. We call those reads spliced. Reads that mapped to introns, that would represent an unspliced message. And ambiguous is a read that maps entirely inside an exon, because we don't know if that's a spliced or unspliced message. As of Cell Ranger version 7, and what we will do today is to make sure that we count all of our unspliced reads as well. I have a few links in the slides here of why we do that. There are a variety of benefits to making sure that we include the unspliced counts in our representation of gene expression. The easiest to understand is we just get more counts. It's a read that is derived from a gene. The data are already very sparse. If we get more counts that represent the presence of that gene, we're gonna be better off. That's the easiest one to understand, but there's a nice like technical note that 10x Genomics released, as well as a few papers that describe this. And so whether we align our reads to the whole genome or the transcriptome or something called a splicey annotation, which is spliced genes plus introns, governs how these reads are counted and allocated. So my preference for data processing is this tool called AlvinFry. It is written and supported by Rob Patro's lab. This is the same group that wrote SALMON, which is a very popular method for bulk RNA-seq transcript quantification. I believe some Dana-Farber people were on that original description. Alvin is within the salmon ecosystem, and this lab, I guess, really loves fish. Alvin is some other like early phase of the salmon life cycle or something like that. This method is really flexible regarding read and barcode geometries and can even allow totally custom methods. So what that means is that it can handle data very easily from 10x Genomics, but also from like a split-seq PARSSPO type of method, or really anything else. Cell Ranger, by comparison, only is going to handle data derived from 10x genomics. AlvinFry can also handle spatial transcriptomic data like 10x Visium or others. It can process hashed or multiplex samples, the HDOs that we mentioned. It can deal with site-seq data. It can do all kinds of other stuff. There's a bunch of helpful tutorials and vignettes on their website. They're all linked here, and they have a very active and responsive GitHub community. The fact that it's open source and it has all of these other criteria mean it's a big win for me, and this is my go-to for processing these days. If you're not a coder and you want to run this a little bit more easily, there are a few helpful wrapper functions or methods to run this. There are two that their lab wrote called SimpleAF, and one is written in Nextflow. It's called QuantAF. Those might be useful to you running in the cloud or something like that. And then NF-Core is a consortium that writes a bunch of centralized pipelines for all kinds of methods, bulk RNA-seq, ATAC-seq, whatever. And they have one for a single cell RNA, and you can choose to run either CellRanger or AlvinFry based on within this pipeline. So if you're doing this frequently, it's a lot of samples, supporting a lot of labs, you might want to check out some of these other wrapper functions. Okay, so let me pause there and ask if there are any questions in person or on Slack about the first part. And once we get past that, we'll move on to the live demo portion. Any questions in the room?
Well, Jeremy, I have a question. So when you mention all those possible counts that can enter or not, the overall number, you're assuming that basically most of the times the data that you get, the raw data that you get, are not the counts, right? So one of your tasks is to actually get these raw data, whatever that could mean, and actually perform counting process of the different cells, am I right?
Yeah, so to rephrase the question, and hopefully I think the room is mic'd, but just to be sure that those online can hear it as well. The question is about the unspliced, spliced, and ambiguous counts, and in a way, why we're making sure that we account for all of those different scenarios in our eventual data matrix. Yeah, I think if we as an analyst assume that the data were processed in a certain way, we may assume incorrectly. And the CellRanger change for version 7 is rather recent. And a lot of people haven't updated to include that parameter yet. And so you may be looking at data that doesn't have all of the possible reads accounted for, and or the worst case scenario is that some of your samples do and some of your samples don't. So that would be like, worst case scenario is that not all of your data are being treated equally. So it's sort of a general philosophy of mine. If I'm receiving data, I want raw data. I want to do it all myself. Maybe I'm just selfish in that way, but I want to make sure that I am positive that everything is treated equally. I'm using up-to-date references, constant, static libraries, everything. So I think it's just the safest bet to make sure, or whoever you're working with, you can make sure that they're doing those steps on your behalf. Does that answer your question?
Yes.
Okay. Okay. I'm going to move on for the sake of time, but feel free. There's going to be a few other moments where we can answer some questions as we go. So the example data that we're going to work with today are peripheral blood mononuclear cells. There's about 1,000 of them. This is a data set that is provided by 10x Genomics. And I can, this link here at the very bottom is on the GitHub link that I shared at the very beginning as well. So it's 1,000 blood cells from a healthy donor, sequenced using 10x version 3, 3' chemistry. It's sequenced to approximately 54,000 reads per cell. And the way that this library is set up is that it's paired end sequencing. The second read, read 2, is the cDNA. So it's like 90-ish base pairs of just transcript sequence. All of the barcode information is on the first read, R1. And so those reads are 28 base pairs, the first 16 of which is the cell barcode, followed by 12 base pairs of UMI.
This is specified by the platform.
This is specified by, so when we say version 3 chemistry, that's what that means.
You already know.
We already know that it's
Where?
it's 16 and 12 in that order.
Okay.
Version 2 chemistry is almost the same, except it's a 16 base pair barcode followed by a 10 base pair UMI. So it is important that when we're processing the data, we're counting the number of nucleotides correctly and determining what our UMI is appropriately. So we really have to know what the chemistry is. Version 3 chemistry is not really new. It came out several years ago. So most samples processed these days are going to be version 3, with the big exception that 5' libraries, as opposed to these 3' libraries, are version 2 chemistry for whatever reason. So if your data are 5' derived, especially for those who do RNA plus TCR clonotyping analyses, you have to know that your RNA data are version 2 chemistry, which means a 10 base pair UMI. We're going to use a few tools today, and the links to the documentation as well as the code repository are all here in the slides. They're also on my GitHub links as well. And hopefully everything is described so that you can get these installed if you need to. For those of us here who are in the data science department, we have Salmon and AlvinFry already installed and ready to go on Kraken as modules. So you just need to do module load Salmon or module load AlvinFry. Okay, so I think to start, we should all make a new working directory for today's workshop. We're going to type at our command line, mkdir, make directory, call it whatever you want. And hopefully you're already downloading or finished downloading the reference genome and gene annotations. And then download these FASTQ files in this format. And then we will decompress those, and then we're going to look at them. So I'm going to pause here for a second. I'm going to show the code on my GitHub for how to do this. I'm going to put this link in the chat as well, just in case. 10x Genomics may ask you to like register or something, but usually if you just copy this link, this code and link directly, it's going to bypass that. So the easiest is to just go to my GitHub repo and pull this exact command. And what you're pulling is a tarball directory of these FASTQ files. I will paste the GitHub link again to the chat in case you missed it earlier. Does everyone in person have that available to them as well? The GitHub link?
In the chat?
In the chat, yes.
If you have to send. Otherwise, we can provide it.
Yep. Maybe those on Zoom, give me a thumbs up reaction or something once your download is complete. those in person, you're welcome to give me a thumbs up as well.
Pretty good fun.
Is yours still going?
4 minutes.
4 minutes? Okay. While that's going, how about I give you a sneak peek at what these files look like and kind of get you oriented. So as I mentioned, the sequencing is done as paired end. And so the R1 file contains the barcode and the UMI information, and the R2 contains the cDNA. So here's just at the very, very top of one of the R1 FASTQ files that we're downloading right now. If you've never looked at a FASTQ file, let me just quickly tell you that each read is a set of 4 lines. So there's a header line. This almost literally gives you X and Y coordinates on the flow cell that the sequencing occurred, as well as some other identifier information. The second line is the actual read sequence. Third is plus sign. And the fourth is an ASCII encoded quality score metric per base call. So sometimes we're more confident based on the imaging process that the sequencing is occurring. We're more confident, so it has a higher quality score. Sometimes there's weird other things that happen, and so we're not very confident. So each base gets a quality score on the scale of 0 to 40, 45. And it's encoded in this kind of weird alphabet. You can kind of look up what each letter or character corresponds to. You can see the very first base on some of these is an N. The quality is different for those. This is probably just because we're at the very, very top of the file. If you scroll down, you'll hopefully see fewer N's. But the first 16 bases so I think if I count it right, that would be these nucleotides that I highlighted would represent that particular cell barcode. And the nucleotides that follow would be the UMI. And then because this is paired, if we look, that means that the R1 and R2 files are exactly in sync with each other. So these are the cell barcode and UMI corresponding to this transcript sequence. So this is 90-ish nucleotides of cDNA that corresponded to that particular cell barcode. And then the second read, also same deal, has a cell barcode, a UMI, and the cDNA that came from it. You'll also see in the files that are coming in your download and often in real life too, these libraries are prepared where the cells are isolated in the same inlet, but then you sequence 2 lanes worth of reads. And that's not really a replicate, it's just to increase our sequencing depth and the number of cells that we can detect. That's denoted usually by this like L001, L002, and so on. In most cases, not necessarily all cases, but most cases, you can consider these as ready to just concatenate. So you just concatenate your L1 and L2 R1 files, and that makes a new collection of all of your read ones. And then you concatenate your L1, L2, R2 files, and that's going to be your collection of R2 reads. We don't have to physically do that. When we run AlvinFry, we can just specify all of the files with a wildcard character that I'll show you in a minute. Okay, are people's downloads complete, hopefully, or almost there? Okay, I'm gonna keep moving for the sake of time. Again, all of this is here, so if you get stuck on any of these steps, you can hopefully catch up or catch up a little bit later too. So the next step on my markdown document is to do the thing that we hopefully already did at the very beginning, which is to retrieve our GenCode annotation in GTF format, as well as the reference genome for HG38. So those downloads hopefully completed while I was talking about single cell platforms and whatever. You will need to unzip both of those files. So these are gzip compressed. So at the command line, just type g unzip for both of those, and that should just take 30 seconds or so. While that's going, I'm going to just talk about this splicey annotation that we're going to generate. So we'll use a tool called Pyro. This is a Python package that you can install with pip or with conda. Should install pretty easily. It's a pretty simple package. There is a way to do this in R as well, if you prefer. They have an R package. I think it's just called ROE, R-O-E, and you can do this same process in R if you prefer. What it takes as input is the reference genome FASTA, the GTF from GenCode, a read length parameter, which is what we expect our cDNA sequences to be. So ours are 90 or 91 base pairs, so we specify 90 here. If you're using some other platform, PARSSPO or something, this will probably need to be changed. And what it's doing is using the annotated splice junctions, it's going to make a reference sequence that contains all exons plus introns so that we can figure out which reads came from the spliced, unspliced, or ambiguous regions of those alignments. If you don't want to install this and make this, or you're running out of time or whatever it is, I do on my Dropbox, which I linked here, I pre-made a version of this splicey index that you can download. It's large. It'll probably take about 20 minutes to download. So just keep that in mind. But you can temporarily skip, if you need to, this Pyro make splice plus intronic step if you need to. If you're working on a computing environment like Kraken, you can submit this job using the Slurm scheduler just by wrapping it in an SBatch, or you can write your own SBatch script if you prefer. But this is the general process. We say Pyro, make splice plus intronic with the FASTA, the GTF 90, and then we give it an output directory name. So I say GenCode v45 annotation splicey underscore FL85. And this is because there's derived fragment length that is 5 less than our read length. You can read more about the Pyro method and the reason why we do all of this. There's a variety of papers that I linked previously. Does everyone have Pyro running? Any questions from those on Zoom? I can put those in the chat and I can try to field those here. Yep.
this is all 3', right?
The example data set is 3'. Yep.
Probably a dumb question, but aren't all the reads are at the 3', actually at the 3' end?
Most of them. Most of them will be.
So you would expect there to be not that many splicing incidents at all, right?
Yeah, generally,
Most genes, I can refer that to the GTF part too.
generally the frequency of a read spanning a splice junction when the library is 3', you can think, I don't know exactly how frequently that occurs. But if it's a 90 base pair read, then yeah, you might, yeah, I'm not sure how often that happens, but there's going to be cases that are definitely a mixture of those 3 buckets, basically.
That are enough to affect the conclusion.
Right, and the poly A selection isn't 100%, and so there's going to be some random events that happen.
Second question then is, not directly related, but how high how many amplification cycles do you do? What's the kind of limit of the UMI counts before deduping in a sample like this one?
Is your question about like how many cells could we get before our UMIs are no longer unique?
No, I mean, I know the answer. I mean, but yeah, but the question is how. No, not the barcodes, but just the amplicon UMI, the 12 nucleotides. So but as that drifts up, I think there's some kind of funny effects that happen about.
You mean if you sequence the library more deeply, or get more cells, or
No, if you get more amplicons and therefore, you get more, then you'll have better chance of getting thinly expressed. But you'll also wash out top end.
Right. Yeah, I think what you're asking is, if you were to isolate more cells or sequence more deeply, you may detect more cellular barcodes, but at a lower depth per barcode. In other words, you kind of dilute your signal by getting more breadth of coverage, but lower depth of coverage. Is that kind of?
Dedupe it, it's no longer, one is no longer one, right?
One is no longer one? What do you mean?
That's what I think it said. Yeah, 20, you got because of the possibility, because of collisions. I guess you actually get 12, I get to 12 nucleotides, so that's 24 bits. So it's always a very small, you never get close to the bit, right?
Yeah, there is some math that is a little bit counterintuitive about at what point have you sequenced so many cells from the same sample that you are no longer necessarily getting 2 cells or 2 events that have the same cell barcode that are not the same cell. They're actually 2 cells that just by random chance have the same cell barcode sequence. That does happen. My very basic understanding of that is that it follows the same math as the birthday problem, in the sense that it's not just the pure combination of number of nucleotides times to the 4th, but you actually get barcode collisions more frequently than that based on how many barcodes you're sequencing. There is some description of that, and it can get kind of scary, especially when you factor in allowing for mismatches, which we often do. There has been some discussion on Twitter and maybe in some papers talking about the frequency of barcode collisions and how we should be more careful.
I think it was a bigger problem when the old days when we had hexamers. When we had hexamers, we only had.
Oh, sure. If you have a shorter barcode sequence, then your rate of collision is going to be way, way higher. The other thing to keep in mind is that if you have two samples processed completely independently, you may have two cells, one from each, that have the same barcode sequence, but those are not the same cell. They're derived from different tissues, and it's just a random combination event that you got the same barcode sequence. Those shouldn't be combined because they're not necessarily, we know them to not be the same cell. Okay, once you have the splicey reference, the next step is to make a salmon index so that the first step of Alvin can count the number of transcripts per cells using that splicey annotation. So the syntax here is to call salmon index. We supply this FASTA file that was generated from the previous step. It's within the output directory, splicey fl85.fa. We pass the parameter that says these transcripts are formatted in GenCode format. What that means is the names of the transcripts, I think, have vertical pipe characters. So there's more than just the transcript identifier. And this parameter will help match transcript IDs to gene symbols. -I specifies the output index directory that salmon is going to write, and we can process this across a few threads using -P8. Again, if we're using the DS servers on Kraken, you don't need to do this, or you don't need to install salmon, rather, you can just load it as a module first. Then for your salmon index step, you would wrap this command in an S-batch just to make sure your number of nodes matches the number of threads you're specifying. Just to repeat, in case you missed it before, I do have this pre-made on my Dropbox. The link is here. So what salmon is doing to create the index is it's really dividing up all of our sequences into all possible k-mers. I think by default it's using k of 31. So it's just counting how many 31 mers occurred how many times. All possible 31 mers that span the entire splicey reference, so that once we actually go to the counting stage, we can just quickly query that 31 mer index, ask how many times did the sequence occur, and therefore what gene was that derived from. Very basic explanation of what this is doing. It's far more complicated than that. Hopefully that's not an oversimplification. Anyone getting any errors? Who's doing this live? I'll give a quick overview of how these next steps go. The next step goes a lot faster. Alvin and AlvinFry basically runs in 4 steps. There's salmon Alvin, which is really just counting transcripts per cells. Then AlvinFry itself runs in 3 steps. There's generate permit list, which is kind of deciding what is a cell versus what is just random noise. And it does this by trying to count how many UMIs were detected per cell and kind of using a heuristic to decide what looks like real signal versus not. The output of that then goes into AlvinFry collate, which I have to remind myself what collate is doing. So it takes this RAD file, which is what the first step of Alvin is generating. Oh, and it's correcting some of these potential barcode errors. So it's collating by the corrected cellular barcode. So it's like grouping everything together by a corrected cellular barcode that then can be quantified in the final step.
So it's like a natural acid.
Yeah. So this first step where we run Alvin, I'll point out a few things. One is we specify -1 and -2. Those are our R1 and R2 files. And as I mentioned, these example data, and often in the real world, we're going to have data from multiple sequencing lanes that we want to treat in combination. So the -1, we just specify asterisk R1, -2, we say asterisk R2. That will collect both the lane 1 and lane 2 data at once and pass them into this Alvin step. We specify chromium V3 that indicates we're using V3 chemistry so that Alvin knows we have which nucleotides correspond to the barcode and which nucleotides correspond to the UMI. As I said before, the barcode for version 3 chemistry is the first 16 nucleotides. The UMI is then 12 nucleotides after that. Both of those are on read 1. And read 2 contains the cDNA. So we say read geometry 2, 1 to end. That's the whole sequence represents the cDNA sequence. So these things are effectively equivalent. Chromium V3 is a shorthand for specifying barcode geometry, UMI geometry, and read geometry. Similarly, for version 2 chemistry, this is mostly important for 5' based libraries. You specify -- chromium, not V3. Or you can say my barcode is 1 to 16. My UMI is 17 to 26 now instead of 28. And the cDNA read is on read 2, the whole thing. This command also specifies sketch and rad. The most important one here is --rad, so that we report all of our reads in a binary format called a rad file. I'll give you a peek of what that looks like. We don't really have to think about it too much because all of the downstream steps are going to utilize reads in that format. --sketch is somewhat optional, but it's what I tend to use. We're performing a fast sketch-based alignment and reporting the reads as a rad file. This is as opposed to the slightly slower pseudo alignment. You can read in some of the documentation and some of their papers about the distinction there.
You don't specify 5 versus 3' there? So that
Here, no. In the next step, we do. I'll also mention that the Patro lab has an alternate method that you could plug in here. Instead of Alvin, you can run PySem. Slightly different approach, but same net effect for RNA-seq. It's still going to, in some way, pseudo align the reads to our splicey index, report them as a rad file for consumption by AlvinFry. PySem is written in Rust, whereas Salmon is in C or C++, if I recall. And we also include this dump features parameter here. This is optional, but if you want to do some QC on your run using an R package called AlvinQC, it requires this one. So we just include that in all of the code and the output stuff that's on my GitHub repo associated with this workshop, I do have the output of an Alvin QC run formed on this. So you can take a look at what that looks like. It mimics somewhat what a Cell Ranger summary report file looks like. It'll talk about how it decided we have X number of cells and throwing away all of the rest of the obvious debris. It'll show you a cool little elbow plot of how and where it drew that line. It's a helpful thing to just run and stash. And if you downstream encounter any issues where a sample is acting as an outlier, you can always investigate, make sure quality-wise everything looked okay upstream. To run AlvinFry, again, if you're using the DS servers on Kraken, you can just load that as a module. Otherwise, you'll need to install AlvinFry. There's a bunch of different ways of doing that, either via Conda, or it's because it's written in Rust, you can follow these steps instead. This, I just did this an hour ago on my Mac fresh, and so I'm pretty certain that this should work at least in that architecture. You first have to install the cargo, or sorry, install Rust up. It's like the Rust toolchain, so that you can then eventually call this tool called cargo. You retrieve the AlvinFry source directory from their GitHub repository, you build it from source, and then you should be able to just call AlvinFry. You can do it from any directory if you add that to your path variable. Again, all of this will be available later in case you need to install AlvinFry yourself. Let's peek at what is inside. Oh, sorry, there's a question on the chat. Let me answer that first. Okay, the question is, would you be able to speak to Alvin's algorithm for empty droplet removal? I believe they generate a cell barcode whitelist de novo with a knee inflection algorithm versus using a whitelist from 10x and allowing some wiggle room. I have heard that depending on the data set, Alvin's method may over or under correct cell numbers. Everything you said is mostly true. So the default method is a knee-based method, which is the same more or less as what Cell Ranger does. So it's trying to figure out where that knee or elbow is and deciding how many cells to keep versus how many cells to throw away. In practice, I have found some variability sample to sample where the number of cells that it calls by default as quote unquote real are smaller than what Cell Ranger may report for the same sample. Both for Cell Ranger and for Alvin, you can specify a parameter called expect cells. It's like -- expect -cells. And you can just give it whatever number you think might be there. So often we have some information from the lab that processed the sample going into the 10x machine, and they will have a pretty good estimate on like, we expect, I don't know, 5,000 cells or 8,000 cells or something. I would give that number here, and then you can specify it for every sample in your experiment. It will, even if it overcounts, that's better than undercounting. If you have more data that you want to filter out later, that's okay in my book. We can set our filtering criteria in the downstream analysis stage to discriminate what we think is a real versus non-real cell, rather than using the upstream heuristic. So that is something that I often do is run these steps with an expectation. Then once we merge and deal with all the data, we can QC and filter out what is potentially noise or debris at that stage rather than this one. But the other part of this comment is that AlvinFry does not use a built-in permit list. And that's why the very first stage of AlvinFry is to generate that permit list de novo from the data. Cell Ranger will, I believe by default, match to an expectation of all of the possible cell barcodes that could be there. There are pros and cons to doing it this way. My understanding is that there can be very real events that are not in the default permit list from 10x, and we would otherwise be throwing those potentially real cells away by forcing it to be in that permit list. So yeah, in my opinion, it's better to generate that permit list de novo from the data, treat all the data the same. You can always filter it out if it is noisy or producing very few reads later on. Does that answer your question, Jennifer? Cool, thank you. So I'll just show you a quick peek of this RAD file. It's kind of like, it has elements of a BAM file. There are some tags here, ID, HI, NH. Honestly, I don't keep track of what any of those are. CB is the cell barcode. So these are the cell barcode sequences and the matching UMIs for that particular read. And it gives you the transcript identifier that read and UMI are mapping to. And so this RAD file is very, very long. It's in binary formats, which means it's not human readable by default. You have to use AlvinFry view or some other tool to view this binary format in a human readable way. So it's often many, many gigs and many, many lines long, but here's just the top little snippet of it. If for some reason you have a BAM file that you want to turn into a RAD format, you can also do that with the AlvinFry convert command, which converts BAM to RAD. I suppose if you really wanted to align your reads using star or star solo or some other tool, you could do so and then convert it to this format and then do the quantification steps with AlvinFry. I guess that would be the use case there. Okay, so hopefully those that are running this live by now have finished the salmon Alvin step to get that first stage of output. The next 3 will run in like 2 minutes or less, which is amazing. If you were to run this same sample with Cell Ranger, we would be here until tomorrow. So AlvinFry is much, much faster. So generate permit list is the next stage. The key here is this -D, which is a shorthand for like the expected orientation of the reads. So here we have a 3' based library. We specify -D FW, which is forward. If it is a 5' library, you have to specify RC, which is the reverse column. There is a little bit of information about this in the AlvinFry documentation. I also at one point created a GitHub issue for them to expand on this a little bit, just to be a little bit more explicit about the 5', 3' difference in orientation. There was a very helpful GitHub repository and workflow by this group called the Alex's Lemonade Stand that is all linked in this GitHub issue if you're really gonna dig into it, but they did the full diagnosis and helped me understand what is correct for all of this. So could be worth looking into. The options are really forward, which filters out alignments to the reverse complement. RC filters out alignments to the forward, which is kind of a little bit counterintuitive in their naming. And then there's either and both, which is basically don't filter anything. And related to the question a few minutes ago, we specified -K here, which is the knee-based method. I believe they do have some other possibilities here, including like something like expect cells with a number. or there are some other options that we can specify as well. So this would be really based on what you know to have been loaded and isolated on the 10x instrument. So I wouldn't just totally guess at a number there. It should be somewhat based on known values. I don't know that you really can do a ton of harm by specifying a fairly large number like 10,000 or something, as long as you're doing extensive quality control and cell filtering and all of that later. So I'll just reiterate, 3' libraries utilize -D FW, 5' libraries utilize -D RC. This example data set is 3', so we specify -D FW. So this step generates that permit list de novo based on the data. Here, based on that needy-based method. I'll show you the output of this from the AlvinQC a little bit later. Then we run AlvinFry collate, which takes that output directory as input and then a new output directory as output and a number of threads. So we can thread this across 16 or whatever processors. This should take only quite literally a minute. Here on my repo, if you're running this on the DS servers, I've just given you the sbatch command to submit this to a Slurm scheduler. All these steps have the potential to be computationally intensive. So you could likely run them on your local machine. If you're running them on a server, be sure to submit them properly, not just run them on a logging bin. There's a question in the chat. What does -T16 stand for? So some of these some of these subroutines like collate -T specifies the number of threads or processors or CPUs that we can run this process in parallel. So as an example, this collate step here, I'm running this on 16 threads. My sbatch command also matches with 16 CPU processors so that this gets properly threaded and run in parallel. You could just run it as -N or -T1. It'll just run more slowly. If you're running it on your personal machine, maybe run it with 4 or 8, but probably not more than that. This will depend on your architecture though, especially if you're running this on a cloud instance or something like that. You're gonna build your instance with a certain CPU architecture, so you may only have a certain number of threads available to you. That will all depend on what your architecture is. The last step of AlvinFry is to run AlvinFry quant. This too runs in just 2 minutes or so. The syntax here is AlvinFry quant, and you specify -M, and we're going to talk about what this file is. It is produced by the splicey, like the pyro step that we ran a little bit ago to generate our splicey reference. This T2G 3 call is a transcripts to gene mapping file that has 3 columns, and I'll show you what that looks like. As input, you give it the output from the previous step, and it writes a new output directory and dumps all of the files that we'll use for downstream analysis into that one. Again, we can thread this, I've specified, I'll run this 16 threads. Again, my sbatch command matches. We specify use MTX, which is a matrix output format. I believe this is the default anyway, so this is just to be explicit about that. The only other parameter to really mention is -R. Your options here are CR-like or CR-like EM. And on the slides, I have a little bit of a description from their documentation. So CR-like is like that in Cell Ranger, that's what the CR stands for, except that it does not first collapse one edit distance UMIs. Within each cell barcode, a list of gene UMI count tuples is created. If a read maps to more than one gene, then it generates more than one such tuple. So all this comes down to like a read that is a potential multi-mapper and how it ends up getting counted or not and allocated in the case of equally good mappings. So this first method, CR-like, if there's a tie for the highest count for this highest count gene for this UMI, the corresponding reads are simply discarded. So maybe not the most ideal approach, even though that's what Cell Ranger is likely doing. But you know, I like to retain all of the reads if possible. So their group developed a spin on this, which adds in an expectation maximization algorithm so that the counts for each gene are determined in a statistically like they're allocated correctly or based on the model. So I typically run CR-like EM, but either of these may be appropriate depending on how you think these should be handled. I believe the AlvinFry paper describes some pluses and minuses to these as well. I mentioned this T2G mapping file. So here's just a little snippet of what that looks like. Again, this is created by the Pyro step that we ran earlier. The three columns are a transcript identifier, which usually begins with ENST, the gene identifier, which is ENSG, and then this third column contains an S or a U or possibly an I call, and it's kind of describing the spliced, unspliced, ambiguous possibilities for these mappings. So we just need to pass the file that Pyro created, this -M flag here. Our output from AlvinFry quant is a directory, and that directory, at least based on this, you can name it whatever you want, but I named it _quant_CR-like EM, so that I know I ran AlvinFry using this method straight in the name of the output directory. That directory contains files that are organized in various subdirectories. There's some metadata, there's some information about how AlvinFry was run, the number of mappings, and so on. In the Alvin subdirectory, there are 3 files, and this mostly matches, or exactly matches, what Cell Ranger produces. So there's one file that's a .mtx file, which is a binary matrix, the actual data matrix of all of our counts. And then there's 2 other files. There's quants.mat rows and quants.mat calls. And that is literally just a text file of all of the cell barcodes as rows or genes as columns. And so if I just do a word, like a line count on this quants.mat rows file, it tells me that Alvin called 1,152 cells. The rest were determined to be noise based on the knee-based method that we talked about earlier. These aren't necessarily high-quality cells, and I'll go through in the last 10 minutes an example of some of the downstream steps that we would begin at this stage. But at least as a starting point, we have about 1,100 cells that Alvin has counts associated with it. At this stage, we move to R. You could do this in Python if you prefer. There's a variety of packages that can read the AlvinFry output. Those are described on the, I believe it's in the Pyro package. There is a method there, but potentially in ScanPy or some of these other Python-based single cell ecosystems, you can import and handle data generated by AlvinFry. In R, at least, we can run this AlvinQC package. The main function that we'll call there is AlvinFry QC report. And we basically just give it all of the directories that it created. We give it a sample ID, an output format. We say we want to see all the code. And I'll show you what this looks like in just a second. But to actually read in the data into R and do something with it, we use this package called FishPond, developed by Mike Love's lab. And its function key for us here is load-fry. And so we give it this CR-like-EM output directory, and we have to specify an output format. And output format here is described at length in the documentation. But the key is either scRNA or snRNA. And this comes down to how we want to deal with those spliced, unspliced, and ambiguous counts. That was the whole purpose of us doing the splicey index to begin with. So if we want to make sure that we're counting everything per gene, then we specify snRNA or U plus S plus A. If you're doing some complicated RNA velocity measurements, there are other ways of doing this. And so you can generate R objects based on the velocity or scVelo or some other combination of these parameters specifications here. But for our purposes, we'll just be specifying output format equals snRNA. Because in 2 weeks or so, data science will be hosting a follow-up to this workshop, which is more advanced single-cell RNA-seq analysis, integration, clustering. What I'm going to say in the last 5 minutes today is really just a very, very broad overview of how we dovetail coming out of AlvinFry into the typical like Seurat workflow. And I'm going to move pretty fast just to give you a general overview of what that looks like. But the more detailed, proper QC, clustering, integration, all that stuff is going to follow in the next workshop. So if you haven't seen that announcement, head to the data science website. There's a whole list of training modules that are upcoming, and you can register for that one in about 2 or 3 weeks. Let me show you real fast what the output of AlvinQC looks like. And then I'll wrap by just showing you real fast some processing code so that we can look at some of our cells and clusters. Okay, this is the output of AlvinQC. It gives you all of the code, which means if you really wanted to, you could run these steps individually once you install the AlvinQC package. It gives you a bunch of helpful metadata to track, especially when it comes to time of writing your eventual publications. You can keep track of your salmon and AlvinFry versions, the index you used, the names of your FASTQ files, and some other parameters along the way. So very helpful thing right up front. Tells you how many reads were processed, number of observed cell barcodes, which is really like any possible cell barcode that had any number of reads. A lot of that is going to be junk. So we know that there's only 1,000 cells here, but we detected more than 600,000 barcodes. We're going to filter a lot of that away when we generate this permit list. In this case, we called about 1,100 cells, and fraction of reads mapping in those barcodes was about 93%, which is quite good. We used the knee-based method, which is trying to determine where that line is between cells and debris based on the cell barcode frequency and the rank. So a decent experiment should look something like this, where you have a sudden drop in this curve, kind of sigmoidal in shape, and where that knee or elbow falls is usually a good indicator of where our line is for determining what was real cells. There are some other helpful plots, number of detected genes, some other summary stats. All of that is included in the AlvinQC output. So I recommend running this for each sample, just stashing it away. You can look through it, especially if there's any technical issues that arise on a given sample. So we use load-fry to load in our AlvinFry output. We specified snRNA, meaning our counts are based on the spliced, unspliced, and ambiguous alignments. And now this is a single cell experiment object. So this package in R is also required, which means that you can look at the counts assay by saying counts and then the name of your object here. And here I'm just looking at the first 5 rows and columns. So here this tells us it's a sparse matrix. The cell barcodes are the columns. The gene identifiers are the rows. If we just do a dim on that, we see that we had 63,000 genes and 1152 cells. I personally like to work with gene symbols rather than gene IDs. Most biologists do as well. There's a number of ways that you can do this, but here is some helpful code that I have written that does some matrix math to map gene identifier to gene symbol. What that requires is a mapping file of the gene IDs to the gene symbols also, which is created by the pyro step that we ran earlier. So this is literally just a 2-column file of the transcript IDs in one column, the gene symbols in the other. And we do some funky matrix math to get a summarized table, which now looks like this: gene names by cell barcodes. There are some duplicated gene symbols, and that's because of the pseudo-autosomal region on chromosome Y. There may be some other weird edge cases, but this will take that into consideration. If you literally just did a lookup, you would have duplicate row names as a result. So it's important to manage that, summarize it. Okay, so now we do a dim of our summarized object. We have 61,000, so slightly fewer than we had earlier. Now in gene symbols by the same number of cells. And now we can create a Seurat object based on that counts matrix. We can count our mitochondrial contribution using percentage feature set. All of this is from the basic Seurat vignette. So we count our mitochondrial contribution, store that as a metadata column, And then we can subset our cells based on a minimum number of UMIs detected, a min and max number of genes detected, and the percent mitochondrial contribution. For full disclosure, my preferred method here isn't to just set a hard cutoff for mitochondrial contribution. I prefer this model called MyQC, which is linked here. So it takes the percent mitochondrial on a cell-by-cell basis and decides which cells should be kept or discarded based on too much or too little, or really too much mitochondrial contribution. So we subset based on those quality control criteria, normalize, scale our data, run principal components analysis. I report at least 100 PCs and then plot that as an elbow plot. And here we can decide how many PCs captures the most variability in the data. So this is reducing the number of dimensions from the total number of genes, which is on the order of 60,000, down to, in this case, 10 or 15 or so principal components, which then is used in the downstream steps where we find nearest neighbors, find clusters, plot those clusters in UMAP space if you're a person that finds UMAP useful. And then you can label those, each point as a cell in UMAP coordinates, colored and labeled by the cluster that it was assigned. Now we can visualize known key genes and their expression patterns in these cells using something like feature plot. Seurat also has methods for plotting as a heat map or a violin plot or anything else that may be useful. Here's a violin plot of those very same genes. You can see some strong cell type specific expression patterns. That's pretty much it. So in a couple of weeks, there's going to be a much more in-depth dive into sample analysis using Seurat. This is just the very brief overview of how we take our AlvinFry output and analyze those data in Seurat. So at this point, I will stop and answer any last minute questions. But as I mentioned, everything is available on GitHub. The recordings will be available on YouTube. I am available by email or Slack and can help you get things installed if needed. If you're in the data science department, then utilize our tools as much as you can, because then you don't have to worry about installing stuff on your own.
Thank you very much, Jeremy.
Thank you all.